Bacteria engage in horizontal or lateral gene transfer, meaning their genes are exchanged between cells of the same generation. In contrast, vertical gene transfer occurs when the parents pass genes to their progeny. The three methods of horizontal gene transfer that bacteria employs are conjugation, transformation, and transduction, with conjugation being the most common. Transformation and transduction typically takes place between bacteria of the same or closely related species. Conjugation is encoded by plasmids or transposon. Plasmids are circular DNA sections that replicates independently of chromosomes. Transposon, also known as transposable element or jumping gene, are mobile sections of DNA that can move within or between genomes. Conjugation with plasmid is more common than conjugation with transposon. Conjugation requires direct cell-cell contact. A conjugative plasmid or conjugated transposon is cell-transmissible. In other words, it has all the genes needed to connect with another bacterium and transmit itself to another bacterium via conjugation. We still don't know the majority of gram-positive bacteria achieve the intimate association of the cell surface required. However, in gram-negative bacteria, this typically involves conjugation pilase, also known as F. pilase or sex pilase. The conjugation pilase binds another bacterium, then retract to pull the two cells together. Once a bridge is formed with an opening between the two bacteria, they are now called mating pair. A nuclease breaks one strand of the plasmid DNA at a ORT or original transfer site. The nicked strand enters the another bacterium while the other strand remains behind the donor. They can now both produce a complementary copy of the plasmid, so the DNA is double-stranded again. Now each bacterium has a copy of the plasmid and both can make conjugation pilase. Note that in population of bacteria, a conjugative plasmid and other that do not. Eventually, all cells will acquire conjugative plasmid. The enzyme tropostasis catalyzes cutting and resealing of DNA during transposition. Note that once mating pair are formed by conjugative plasmids or transposon, this also allows non-conjugated transposon and plasmid to be transferred to another bacterium. Transformation occurs when bacteria takes up extracellular DNA to incorporate into their genome. Typically, this occurs when one bacteria lyses or splits open, releasing its genetic content, and then another bacteria comes by acquiring it. Bacteria able to bind large amount of DNA are called competent. Competency is a state of increased cell wall and cell membrane permeability that allows cells to uptake DNA. Many bacteria are naturally competent and so actively bind environmental DNA. After transport into their cytoplasm, the bacteria can incorporate the new DNA into its genome through the process of recombination. Recombination is the rearrangement of the donor and recipient genomes into new hybrid genome. This can result in new phenotype. For example, the bacteria can acquire pathogenicity or antibiotic resistance. Some competent bacteria actually kill non-competent bacteria to release DNA for the transformation. Transduction occurs when the DNA is transferred from one cell to another by a bacteriophage, a type of virus that infects bacteria. Viruses cannot replicate on their own. They are obligate parasites that can rely on a host machinery. Many bacteriophages can switch between a state of lysogenesis and a lytic cycle. When a bacteriophage is in a state of lysogenesis, the virus combines its genome into the bacterial chromosome. The viral genome hangs out there many generations. The bacterium replicates when induction occurs. The virus switches to lytic cycle. The cell becomes virus-producing factory until it gets full of virus, that is lysis, or burst opening releasing virus, particularly into its surrounding. There are two kinds of transduction, generalized and specialized. During generalized transduction, the phage capsid accidentally assembles around fragment of the bacterial DNA or a plasmid. When the assembled viral particle infects a new bacterium, it injects its previous host DNA into the new host. Specialized transduction occurs during the lysogenic cycle of the virus. Incorrect accession of DNA during induction results in the fragment of bacterial DNA being picked up instead of the part of the viral genome, which then stays into the bacterial nucleotide. Now the bacterial DNA replicates as a part of the bacteriophage genome, is packed into the phage capsid and is injected into the new bacteria. Horizontal gene transfer along with mutation allows bacteria to achieve genetic diversity. These processes allow bacteria to adapt to new environment because they enable them to respond to selective pressure. Mutation is a slow process, and most mutations are harmful or neutral, not beneficial to the bacteria. Meanwhile, horizontal gene transfer is a rapid way to acquire large chunks of DNA from another bacterium all at once. So if the bacteria keeps acquiring new snippets of the DNA from another bacterium, do their genomes keep getting larger and larger? No, if the transfer genes don't provide selective advantage, they are usually lost by deletion, so the genome size remains constant over time.